Students, today we'll start with mitochondria. Mitochondria is double membrane bound organelle, right? It was discovered by Colliker and it was named by Benda. Name of the scientist is not given in NCERT, but it is important that it was discovered by Colliker and named by Benda. Now, first we'll discuss about the structure of mitochondria. This is outer membrane, right? This is inner membrane. The space between two membranes is perimitochondrial space. Right? Now, the inner membrane has few enfoldings. These enfoldings are termed as crista. Right? Now, why crista is present? Why enfoldings are present? To increase the surface area of inner membrane. Right? These racket-like structure is oxisome. Here we have double-stranded DNA. The dotted structure is ribosome. Type of ribosome present in mitochondria is 70S. Right? And the fluid matrix is mitochondrial matrix. So that is the structure of a mitochondria. Outer membrane, inner membrane, the space between two membrane, perimitochondrial space. Then racket-like structure, oxisome, mitochondrial matrix, crista, ribosome which is 70S and double-stranded DNA. These are the component which is present in mitochondria. Now let us start with few important points about mitochondria, then we'll discuss about structure of mitochondria. Number of mitochondria present in a cell depends on it depends on physiological activity of the cell. Now why it depends on physiological activity of the cell? Suppose a cell is energetically active, then it will have more mitochondria because mitochondria is known as powerhouse of the cell because it generate or produce ATP, which is energy currency of the cell. So any cell which requires more energy will be having more mitochondria. So it depends on physiological activity of the cell. If we compare plant cell and animal cell, then plant cell has less number of mitochondria and animal cell has more mitochondria. Shape of mitochondria. Shape is either it is cylindrical or it is sausage shaped. Right? Size of mitochondria. Diameter is about 0.02 to 1 micrometer, which average makes 0.5 micrometer, and length of the mitochondria is 1 micrometer to 4.1 micrometer, right? Next is stain used for mitochondria. Special type of stain is used for mitochondria, that is Janus green stain, right? And mitochondria are not visible unless it is specifically stained. Unless specifically stained by Janus green stain, it is not visible. Specifically stain are not easily visible. Now, next is two membranes of mitochondria. One is outer membrane and the other is inner membrane. Outer membrane is smooth and continuous, right? It is outer membrane as compared to inner membrane is more permeable. Because of presence of porins, it is more permeable if compared with inner membrane, right? Second is inner membrane of mitochondria. Now, inner membrane of mitochondria has enfoldings, and those enfoldings are termed as crystal, right? So, why crystal are present in inner membrane? These enfoldings are to increase the surface area. Why surface area is increased? Why there is a requirement of increase in surface area? Because the inner membrane has electron transport system which is required for respiration or energy production. Energy production means ATP synthesis. So that ATP synthesis take place in inner membrane, and if we require more ATP, the number of crystal will be more if ATP is required more by the cell. So inner membrane has this inner membrane has racket like structure, that is oxysome. Now, this oxysome have two component. Suppose that is inner membrane of mitochondria. This is inner membrane of mitochondria and this structure is oxysome. This component is termed as F naught component or F1 F naught particle, and this is F1 particle. F naught particle and this is F1 particle. Both forms ATP synthase complex, right? Now we'll learn about the differences of both the component. One is F naught particle and the other is F1 particle. If we compare F naught with F1, this is F naught particle, right? This F naught is completely embedded in the inner membrane. So we can say F naught is trans membrane protein. We have already discussed integral protein or transmembrane protein, which completely penetrate the membrane, and it is responsible for facilitated diffusion. While if we compare it with F1 particle, so this is inner membrane. Outside the inner membrane, we have perimitochondrial space, and this side is matrix side. So F1 particle is protruded out of the membrane towards matrix side, right? So that is the difference between F naught particle and F1 particle. F1 particle is associated with ATP production. during cellular respiration. Now, how it is involved in ATP synthesis? Suppose we have this inner membrane, this is F naught particle, and this is F1 particle, right? Suppose H plus ion concentration is more toward perimitochondrial space. So H plus ion will move from higher concentration to lower concentration. And by the movement of this H plus ions, a molecule of ATP is synthesized. That is how F1 particle is responsible for synthesis of ATP molecule. Right? So that is the function of H1 particle. So there is one special note, that is F1 particle of oxisome is concerned with, there is a term for ATP synthesis, that is oxidative phosphorylation. Phosphorylation means synthesis of ATP. Oxidative means by using oxidation reduction reaction, right? Oxidative phosphorylation, that is formation of ATP. So we can say, since ATP is synthesized in mitochondria, we can say very, very important point for NEET. Mitochondria is powerhouse of the cell. Why it is termed as powerhouse of the cell? Because energy in the form of ATP is produced in mitochondria. So this statement can be asked in the form of assertion reason. Suppose assertion is mitochondria is powerhouse of the cell. That is assertion. Reason is because energy is produced in mitochondria. That would be the reason. So we can say mitochondria is the site of aerobic respiration. We have two type of respiration, aerobic respiration and anaerobic respiration. Site of anaerobic respiration is cytoplasm, while site of aerobic respiration is mitochondria, right? Next component is mitochondria. Just like a prokaryotic cell, they have their own double-stranded double-stranded circular DNA like this. The single double-stranded circular DNA is present in mitochondria. That is a feature of prokaryotic cell. Apart from this DNA, they have their own ribosome for protein synthesis, and type of ribosome which is present in mitochondria is 70S. So these two features are similar to that of prokaryotic cell. Hence, we say mitochondria have endosymbiotic origin. That means it is originated from prokaryotic cells, since they have some similar feature as that of prokaryotes. So mitochondria, since they have their own DNA, they have their own ribosome and some RNA particle, mitochondria is also known as Mitochondria is semi autonomous organelle. Why it is semi autonomous organelle? Because they have their own double stranded DNA which is circular, then ribosome and some RNA component. That is why mitochondria is known as semi-autonomous organelle. Next important point about mitochondria, just like a cell, mitochondria also divide. And what is the mode of division of mitochondria? Mitochondria divides through fusion. Means simply it divides into two mitochondria. So this you have to remember. This may come in your exam. Then how mitochondria divide? Mitochondria divide through simple fusion mechanism. So that is all about mitochondria, a double membrane bound organelle which is present in both plant and animal cell. More number is in animal cell, less in plant cell, but it is present in both plant and animal cell. More mitochondria are present in those cells which is actively synthesizing molecule of ATP or energy rich compound. So that is about mitochondria. Next double membrane bound organelle is plastids, right? Now what is plastid? Plastid is also double membrane bound organelle. But these double membrane bound organelles are present in, these plastids are present in all plant cells, because plant cells are photosynthesizing cells, and in euglenoids. Euglenoids are also photosynthetic in nature, so plastid are present in plant cells and euglenoids. Plastids are easily observed under microscope. Why it is easily observed? Because they are large. Large in size. Now, very important point, plastids have some specific pigments present in them. So on the basis of pigments, plastids are of three types. This division is on the basis of pigments. Which type of pigment is present in these three plastids? So number one is chloroplast. Number two is chromoplast. Number three is leucoplast, right? So first we discuss about chloroplast. Chloroplast are chlorophyll containing green colored plastid. So color of chloroplast is green, color of chromoplast is colored but other than green, right? So main pigment present in chloroplast, chlorophyll is present in chloroplast, but amount of chlorophyll is way more than chromoplast. Chlorophyll also present in chromoplast, but amount of chlorophyll present in chloroplast is way more than chromoplast. Apart from chlorophyll, they have also carotenoids, but obviously content of carotenoid is less in chloroplast. So chlorophyll gives green color to the chloroplast. Why chlorophyll and carotenoids are present? These are present for trapping energy, which is essential for photosynthesis. Basically, it will trap light energy, and light energy is converted to chemical energy for photosynthesis. So function of chloroplast is to trap light energy. Next is chromoplast. Chromoplast is colored plastid, right, other than green. It has fat soluble carotenoid pigments, and these carotenoid pigments can be such as carotene and xanthophyll. If we talk about color of carotene and xanthophyll, xanthophyll is yellow in color and carotene is yellow to orange. Color is yellow to orange. So what is the function of chromoplast? It gives color to the different parts of plant. So chlorophyll should be present or absent in chromoplast. It is either absent chlorophyll, either absent, and if it is present, then in very less amount, right? So if we talk about the pigment, chloroplasts have both chlorophyll and carotenoid. Number of or amount of chlorophyll is way more than chromoplast. Carotenoid is less as compared to chromoplast. Chromoplast have more amount of colored pigment, that is carotenoids, which gives them different color. Next is leucoplast. Leucoplast is colorless plastid. Why it is colorless? Because pigment is absent, right? So we have function of leucoplast. Function of leucoplast is storage of nutrient. Now what type of nutrient is stored in leucoplast, and which type of three type of leucoplast on the basis of type of nutrient stored? We have number 1 amyloplast, number 2 alluroplast, number 3 alloplast, right? So this categorization is on the basis of stored nutrient. So amyloplast store starch. Example of amyloplast is potato, right? Alluroplast store protein. Examples are pulses, right? Alloplast store fat. Example is oil yielding seeds. these are type of plastid which is present in plant. Now we'll discuss about one important plastid which is present in plant. That important plastid is chloroplast. First, let us discuss about structure of chloroplast. Right? So that is double membrane bound structure. Then they have these coin like structure, that is thylakoid. Pile of coin like structure is grana. Two grana are interconnected through stroma lamina, right? That is the structure of a chloroplast. It has starch granule, and just like mitochondria, they also have their own circular DNA and their ribosome. So let's label the structure. This is the outer membrane. This one is inner membrane. The space between two is periplastidian space. The matrix within the chloroplast is stroma, right? That is starch granule where starch is stored. This single coin like structure is thylakoid, and the pile of thylakoid is grana. Two grana are connected through stroma lamellae, right? This is the double stranded circular DNA, and these are ribosome. So ribosome, type of ribosome, just like mitochondria, they have 70s type of ribosome, double stranded circular DNA, and some RNA protein. Because of the presence of these components, chloroplast or plastid is also known as semi-autonomous organelle, right? Now, where these chloroplasts are present? Majority of the chloroplasts are present in green part of the plant. If we talk about green part of the plant, then one important plant part which comes into our mind is leaf, right? So in leaves we have specialized tissue, that is mesophyll tissue, where chloroplasts are present. If we consider number of chloroplast in mesophyll tissue or mesophyll cells, then number can be, it can be 20 to 40 chloroplast per cell. While in Chlamydomonas, which is a unicellular algae, one chloroplast is present per cell. Per cell is present, right? Now, shape of chloroplast. It can be lens shaped, it can be oval, it can be spherical, discoidal, disc-like, or even ribbon-like. So these are the shape of chloroplast, right? Size of chloroplast, length is 5 to 10 micrometer and width is 2 to 4 micrometer, or we can say the diameter, right? But generally we use the term width for chloroplast. Now, next point about chloroplast is the two membrane system. Just like mitochondria, chloroplast also have two membranes. One is outer membrane and the other is inner membrane. Just like mitochondria, outer membrane is more permeable and inner membrane is less permeable. But there is one important comparison on the inner membrane. Inner membrane of chloroplast is less permeable as compared to inner membrane of mitochondria. Now this point is very very important. This statement as it is came into in NEET 2024, right? The inner membrane of chloroplast, if we compare it with inner membrane of mitochondria, it is less permeable, or we can say inner membrane of mitochondria is more permeable as that of chloroplast, because mitochondria has to transport a lot of compounds like NADH for respiration. So inner membrane of mitochondria as compared to chloroplast is more permeable. The space which is limited by inner membrane is called stroma. In mitochondria, it was mitochondrial matrix, but in chloroplast, it is stroma. There are no enfoldings in inner membrane of chloroplast. Now, components which are present in stroma. Number one component is thylakoid. Now, one important point about photosynthesis. In photosynthesis, we have two type of reaction, right? Photosynthesis, we have two type of reactions. One is light reaction. Light reaction means plant will receive the sunlight, means absorb the sunlight, it will convert the solar energy into chemical energy. That is the light reaction, and this light reaction involves photosynthetic pigments, and those photosynthetic pigments are present in thylakoid membrane. So thylakoid membrane have photosynthetic pigment. Because of the presence of photosynthetic pigments, we can say thylakoid or grana is the site of light reaction. We can write here also grana is pile of thylakoid. So if there is given that grana is the site of light reaction, that is also correct. It is also considered site of light reaction. Since for light reaction photosynthetic pigments are present in grana or thylakoid, it is the site of light reaction. Second is dark reaction. Product of light reaction is oxygen and ATP and NADH. Sorry, ATP and NADPH. These are molecule of chemical energy. It is utilized for CO2 fixation. We can say CO2 fixation, or we can say formation of glucose molecule. So dark reaction require these energy compound which is produced by light reaction, and Dark reaction requires enzymes for synthesis of glucose. We can say either synthesis of glucose or we can say CO2 fixation, right? Synthesis of glucose. These enzymes are present in stroma. So we can say that second component, which is stroma, is the site of dark reaction. These 2 points are very important. What is the site of light reaction? What is the site of dark reaction? Site of light reaction is either you can say thylakoid or you can say grana. But site of dark reaction is stroma because enzymes for carbon dioxide fixation is present in stroma. Now, what are the other component which is present in stroma? We have double-stranded circular DNA, then ribosome which is 70S and some RNA molecule, right? Because of presence of these component, we can say chloroplast is also semi autonomous organelle, right? Now, function of plastid. Number one, photosynthesis. Or we can say biogenesis. Next, one is important note about just like mitochondria, plastid also have endosymbiotic origin from prokaryotes, that is cyanobacteria. Since they have some feature similar to prokaryotic cells, so we can say they have endosymbiotic origin from cyanobacteria, right? So these two are important organelle, which is double membrane bound organelle, right? In next lecture, we'll be discussing about a non-membrane bound organelle, that is ribosome, right? We'll discuss ribosome, which is non-membrane bound organelle. Another non-membrane bound organelle is endoskeletal system or cytoskeletal system. We'll discuss about these non-membrane bound organelle and one membrane bound organelle, that is microbody. And we will discuss about all these components in next lecture.